All right, are we ready to go? Well, 30 participants, great. Welcome everyone to this new series of seminars from Bioconductor. We will be carrying these out by Zoom, hopefully every 2 or 3 months. I'm very excited to have Wolfgang Huber, Constantin Ahlmann-Eltze, and Simon Anders here as our first presenters, and Davide Risso, who will be the discussant. The format of these seminars, we're trying to engage some amount of discussion, which is very hard on Zoom, but what we're hoping is that the presenters will take about half an hour to present their paper, and then the discussant will have about 10 or 15 minutes to raise some issues, which the presenters will be able to respond to, and then hopefully we'll have about 5 or 10 minutes at the end in order to take questions from the audience. And please make sure that you put your questions in the Q&A, not in the chat, if you have some, and then we'll try to bring those up at the end of the seminar. Thank you very much. And Constantine, I think we're over to you.
So right, in fact, before Constantin starts going into the technical points, I will give a sort of very sort of high-level conceptual framing of what we're doing here. So I'm Wolfgang Huber, one of the authors of this work. So it's about benchmarking. Benchmarking is in some sense related or trying to measure quality of algorithms or of products. So we're comparing different products, in this case, different foundation models, and want to find out which ones are like how good they are and how they compare. So it's really a question of quality. And now if you think about what quality actually is, obviously we're not the first people to ask that. And there are many different definitions of what quality is, and this may probably, and some are quite orthogonal in some respect. And two of the main directions there are adherence of a product to specifications. So how well do, for instance, the angles and the shininess of the surfaces and the precision of how it's being put together adhere to the specs. The other one is fitness for purpose. And that is probably in many cases more relevant to what we mean by quality. The point is that it is a very subjective thing. So obviously you can see I like car analogies, and fitness for purpose can be very different depending on whether you're sort of by yourself on a road trip across the continent, or whether you're trying to get 4 kids to sports lessons. And in some cases, you will need some very different things. And also the adherence specifications doesn't necessarily mean and need to be correlated with the fitness for purpose. There's also this possibly apocryphal quote by Henry Ford. If I'd asked people what they wanted, they'd have asked for faster horses. So obviously, it's not, people don't even often know what they want, which makes quality assessment even harder. So next slide, please. Another thing, so we're measuring quantities, and actually we will come back to this afterwards with specific instances of that. So it's actually quite fun to see this law at work, also in our area. So Goodhart's law, this was an economist who said that, but in any case, when a measure becomes a target, it ceases to be a good measure. Can you maybe add the, right, so here's a sort of fun example of that, how to improve your teaching evaluations without improving your teaching. So metrics, measures, so often metrics that we use are correlated with what we actually mean. And the correlation can be more or less good, but the problem is if once people figure out that, they may also be able to hack the metric and improve the metric without actually improving the thing that we actually want to measure. Next slide, please. So benchmarking is really difficult. There is not even ground truth, and this is something we can come back here as well. Well, in perturbation prediction, nicely, there is in some cases ground truth. In other fields, like for instance, annotating cell types, that is debatable. In a foundation model, again, there's also no ground truth. Then there's this sort of cliche, all models are wrong, some are useful. So how do we define usefulness? but what is actually usefulness. And now with these sort of philosophical remarks, I pass over to Constantin, who will give them a lot more meaning.
Yeah, thank you very much. Yes, so I'm going to kind of go over this kind of work where we kind of benchmarked different foundation and deep learning models for perturbation predictions. And what basically caught our eye was that there were several of these very high impact publications and very high visible journals saying that they kind of were able to perform these perturbation predictions. And what do we actually mean when we talk about predicting the effect of unseen perturbations? Because there are many different things that you can measure about a cell. So you could look at the cell morphology, if it kind of survives or dies, the metabolome, but we will really specifically focus on the transcriptome as the main readout because it's neatly accessible. And then also for unseen perturbations, there are also, again, different ways that you could look at these, but we're going to particularly focus on unseen double perturbations and to some extent also unseen single perturbations. And the idea is in some sense relatively straightforward for double perturbations. We have our perturbed cells here in blue. Then we, let's say, train data on the green and the orange perturbations. When we get the kind of corresponding cell states. And now we ask if we give the green and the orange perturbation together. So if we knock out these two genes, what will the state look like? And it will be somehow a mix of the green and orange state. And the question now is, can these models figure out how these two things specifically mix and kind of make good predictions? And for our benchmark that we kind of worked on, we used the data set by Norman et al. Because at the time that was the only data set really available. I think also pretty much still is the only data set that's publicly available. It's from a cell line where they use the CRISPR activation system. So it's actually not a knockdown, but the CRISPR guides are overexpressing the particular target gene. And there were 101 single perturbations and 60 double perturbations. And our kind of setup was that we always used all the single perturbations plus half the double perturbations for training, and then evaluate on the other half of the double perturbations. And these were the different models that we looked at. So as I already mentioned, there were these kind of papers by SCGPT, SC Foundation, and Gears, which kind of like 1 or 2 years ago came out and were saying that they now had this new framework to be able to actually make these predictions. And we compared them against like CPA, which is another popular autoencoder-based baseline, and 3 other foundation models, which didn't actually themselves claim that they would be able to perform this perturbation prediction task. But because we were curious if the kind of learned representation would actually be good for this, we added a linear decoder to see if the embedding that they are producing can still be used. And then we compared them against some very simple baselines where for the double perturbation task, we looked for perturbation A, the orange one, and the perturbation B, the green one. And our combined perturbation prediction is simply taking the difference compared to the baseline with our perturbation summed up, which we call the double perturbation or the additive model. And what you can then do is we can have a look at, for any particular gene, we can look at the prediction of the observed and the model prediction. So we can take, for example, the Gears prediction. And then if we do this overall, we can see that the more highly expressed genes are still more highly expressed. And we can calculate the error, which is the difference summed up across all genes from the predicted and the observed values. And we can also calculate the R² values. But as you can already see, if we do this for each gene at the full expression, that the R² values are all pretty close to 1. So it's actually more interesting to subtract the baseline. So to basically only look at the length of the arrows compared to the blue line, because then you can already see that there's quite a few differences between the methods. The overall error doesn't actually change because that is unaffected by if you subtract the same thing on the left and on the right, but the R² value actually is changing dramatically. And so here I'm showing the kind of all the readout genes, so like 1,000 readout genes for one particular double perturbation. And if we now don't do this across all the different double perturbations, which we have in our test set, we can kind of quantify how well each method actually performs. So we do this with the random seed of kind of 5 test training splits. And then these are all the readouts. So in orange, I'm highlighting the gene that I just showed you. And when we kind of look at this, the thing that really stands out is if we look at the prediction error as a readout, the additive baseline performs much better than any of the other models. And to kind of confirm that this wasn't just something due to the metric, we also kind of looked at the correlation coefficient using this Pearson delta, where delta means that we're looking at the correlation coefficient after subtracting the baseline, and again, finding virtually the same result, that the additive baseline is clearly outperforming the other methods. Another question was, how many genes should you actually consider for this calculation? So you could, for example, only look at the top 100 most expressed genes. You could look at all the genes. We decided to look at the top 1000 most highly expressed genes. But this graph basically going from kind of along the kind of number of genes that we're considering shows that the results are pretty stable across the range. People also like to actually separate these by look at them by differential expression, where we again find the same type of pattern for these double perturbation predictions. When we first got these results, we were quite surprised because we didn't actually expect the result or the kind of difference between to be so strong. So we decided to actually approach some of the authors of the original publications and ask them what was going on, because it can easily happen that when you try to set up such a benchmark that you make some user error, that you're calling the methods where you miss some suboptimal tuning parameters or maybe miss something. So when we reached out, one of the responses that we got is that maybe this is not really the right thing to look at. So maybe there's a difference between trying to make a good prediction across all the genes versus trying to identify non-additive genes. So next we set out to actually check if these models are useful for identifying these non-additive effects. So the way that we kind of quantified this or operationalized this is again that we have these kind of two individual perturbations, and then we define something around the additive range. So we can calculate what the additive prediction would be, and then we identify some error around that. And if it's kind of between the baseline and the additive prediction, that would be like a buffering interaction. Otherwise, if it exceeds it, it's synergetic. So what we did in the end to actually really find out how wide these bars should be is that we looked across all the readout genes. What is the difference between the additive prediction and the actual observations? And this follows along this kind of bell-shaped curve. And we then decomposed this bell-shaped curve into like a null distribution based on a normal assumption. So this is shown in red. And then we have these wider tails, the wider shoulders in black. And we can find the lines shown here with the black vertical lines, which separate out left and right the regions where we are more than 95% sure that these regions, the kind of genes in here, do not come from the kind of null distribution that we have here shown in red. So we have this roughly 5,000 genes from our total 120,000 readout genes. So these are all the readout genes across the different double perturbations across 5 different test training splits. And then we can, for all the genes, stratify them by what is actually the prediction of the model. So here, this is the same x-axis that we had for this histogram, but now we're in the y-axis for each gene, readout gene. We're also plotting the predicted expression here, taking the SGPT as an example, minus the additive expectation. And ideally, we would want all the predictions to be along this diagonal line. Of course, this is difficult. And so what we do is we kind of look how often do these kind of genes occur outside this middle stripe. So inside the middle stripe is the additive, which we consider the truly additive examples, and outside are the truly non-additive. And then we can count how often these are truly non-additives, the prediction from SCGPT. So we basically work our way down for the most extreme non-additive examples. So we use the examples where SCGPT predicts the largest difference from the additive baseline, and then check if those are true positive or non-true false positives. So we can kind of work our way down this list and then kind of count up that the first and so on are true positives, and then calculate also the false discovery proportion. So that for a fixed false discovery proportion, we can then quantify what is the fraction of true positives that SCGPT recovers. And we get this line. But now again, we need some baseline to actually establish if this is a good prediction or if it's not so useful prediction. And the baseline that we used in the first instance, which was this additive model, isn't particularly useful here because if you, from the additive prediction, subtract the additive model, you're just getting 0 everywhere. So this is completely useless for ordering the genes. But we can look at the no change model. So in the no change model, we just predict the expression that we saw in the unperturbed cells for all the different genes. And then we can make basically the same type of picture here. And when we again quantify this, going down, we find that we get a larger true positive rate for the same force discovery proportion with a no change model than that we got with SEGPT. Basically meaning that just predicting no change is more useful for recovering truly non-additive genes than trying to do this with SGPT. We ran this across all the different foundation models, and the pattern stayed the same, that the no change model performed best across all these different across all these different methods. So meaning that these methods don't actually do a better job of identifying non-additive examples than just predicting no change. One small caveat here is that the no change baseline is always predicting buffering interactions. So it never predicts any kind of synergetic ones. But we also kind of looked at this in the details of the paper. But basically the result was that also these models quite rarely predict synergetic interactions, and it's even rarer that these are correct. So one of the other things that the models are advertising as being particularly effective for is predicting the effect of unseen gene perturbations. So the idea is there that we kind of have, again, the base states, and now we're trying to predict the state if we see some new black perturbation, let's say. And the easiest thing that you could predict is just to predict the mean across all the seen perturbations that you have in your training example. So this is one of the baselines that we're going to use. Alternatively, you can also try to figure out if you can somehow relate the gene perturbation, the black perturbation, to any of the other known perturbations. So if, for example, you know for the gene expression that the black gene is somehow close to the kind of green genes and further away from the orange and the blue genes, then you might also predict that for the space of perturbations, the black perturbation might be closer to the green perturbations than the orange and blue perturbations. And then you would predict that maybe after black perturbation, the cell state would look more similar to the green cell state. We kind of went through the same kind of mechanics again, looking at 3 different data sets. And here I'm showing the results for the Repogler K562 data set. And what you can see here is that, again, for the prediction error, just predicting the mean across all the different seen perturbations performs as good as the predictions from any of the other foundation model or deep learning models. Again, saying that the models don't outperform these baselines, meaning that they don't show any evidence of having learned additional biological relevant kind of gene regulatory networks. We also kind of tested the same on the Repogler RPE1 and the Adamson dataset, and again found basically the same results. What we did find, which did make a difference, was if you try to train this linear example that I gave earlier on one Repogler data cell line, and then try to predict from this one cell line the perturbation effect for the other cell line, that this actually did work and did improve the performance. So this is what I'm showing down here, that kind of this is the only thing that really meaningfully outperformed the kind of mean baseline that we established earlier, suggesting that maybe training on more relevant data is actually a potential way to improve performance instead of trying to learn on these large observational single cell atlases. We were somewhat lucky with this study, so we kind of came out with this a bit more than a year ago, and I think within the week after we kind of put this on bioRxiv, there was a slew of other papers which basically all had more or less come to the same conclusion, coming out on bioRxiv, confirming that kind of this wasn't just kind of fluke from which we found there, but kind of further backing this up and coming to similar conclusions. We published this kind of earlier this year, and you can read it all up in this publication. Now, since then, since this came out, there have been other papers or many papers, of course, are very interested in this topic. I think there are 2 papers which are particularly interesting because what they're saying is that maybe we're still looking at the wrong metrics to quantify if a perturbation prediction is actually any good. And I want to particularly talk about these 2 papers. One is the Systema paper and the other one from Miller et al. And to quickly kind of give you an overview what these papers talk about, the basic idea of this thema is that there might be some general systematic effects and you might want to correct for them. And one metric that they suggest is to look at the overall centroid accuracy. So you look, if you're trying to predict this purple cell state, so this is shown with this dashed line, and you make this particular perturbation prediction, you would then look along your list of all the different seen perturbations, how many of the perturbations which you didn't try to predict are closer to the one that you actually did try to predict. So you're kind of looking at this distance and then count how often the kind of distance of the prediction to any of the other seen perturbations is actually shorter than this one to the true perturbations. And suggests that this could be a useful metric to look at. They also have this additional metric that they're suggesting, where they saying that if you're calculating this Pearson delta correlation score, so here's some pseudocode illustrating how the Pearson is correlated. So this is the first one that I showed you, where basically all the methods had a correlation of 1. This is the regular Pearson delta, where you subtract the mean from the control cells. And they're now suggesting that maybe we should subtract the mean from the perturbation, from the observed perturbations, so that you avoid this effect where the control cells are somehow outside the kind of distribution of perturbed cells, and then you kind of get a maybe more meaningful Pearson delta score. The other paper by Miller et al., I think, has a really interesting idea where they suggest not to just look at negative examples, but also look at positive examples to try to define like a positive baseline. And the idea behind this is that if you only look at the mean expression, you can't really know if the underlying gene expression is this very kind of low variance predictor where you can reliably tell the difference, or if it's actually this very high variance predictor where the difference might not be as meaningful. And the way that Miller et al. Suggests to go about this, or to identify and quantify and rank different metrics, is to split the cells into half and then see how well each half predicts the other half. And they use this to make this calibration of the different metrics. So they look at lots of different metrics, and they say that metrics where there's little difference between kind of the negative control baselines that we've introduced and this positive example, those are not ideal metrics. And then metrics which get a larger difference, they call those calibrated and say that these would be more interesting. And they basically conclude that an interesting, or one of the metrics that they find to be particularly well calibrated, is the mean squared error weighted by the differential expression. So now we're at this point where we have all these different ideas about what you can actually measure and which type of subset of genes should you measure those on. So you could look at the mean squared error. You have the different ideas for the Pearson delta correlation. That's a centroid accuracy. And you can calculate those across all genes, only across the highly expressed genes, or maybe across the differentially expressed genes. And from my perspective, there are pros and cons to either, to any of these different choices. I would like to particularly comment on two of the, which were the ones that were proposed by these two papers, which is to calculate, for example, the Pearson delta with respect to the perturbation mean. The advantage is that it's robust to systematic changes. I think that's nice. However, I think it's problematic to conclude that the additive model does not outperform the other methods with this metric, because if you already subtract the mean from the additive, the mean of the perturbations from the additive, from the baseline, the model really has no chance to kind of make a good prediction. So if you think about this unseen gene perturbation task, where we kind of predicted as a baseline, there's the mean across all perturbations, and now you're kind of calculating the Pearson correlation after subtracting that exact mean, you're basically forcing this particular metric to be 0. So I think this is why one would need to think very carefully about what alternative baseline one would want to compare against. The other, which I've seen in many papers, and I think it's quite popular, is to look at the most differentially expressed genes as for the set of readout genes to assess how well a particular method does. And I think this has the problem that you have no negative examples there. So a method that would predict some differential expression across all the genes might still actually look pretty good according to this metric, because you're never considering genes which aren't changing. So that's why kind of If I had to choose something, I would go with a mean squared error, and I would focus on the most highly expressed genes because those are the ones for which we can just the way that the counter solution work make most reliable kind of inference of the differences. However, this is still very much thinking kind of inside this box, and I want to kind of open this a bit further up. So inside the box is kind of this thinking to try to find the best metric for deep learning models. And yes, as I already mentioned, I personally would go with a mean squared error on the most highly expressed genes. However, what we should maybe, if we broaden our thinking a bit, what we really care about is, of course, that we building these models and try to do this perturbation prediction to understand actual interesting features about cells. So maybe we want to actually care about more of the proliferation, or we care about the phenotype of T cell exhaustion, or how the contractile strength of a cell changes. And in the end, we would, of course, want to learn this from like not just simple perturbation data on kind of cell lines, but from complex spatial temporal data. So kind of when kind of building these benchmark models, always keep in mind that one has actually this much broader interest. And with this, I want to come to the end of my talk. Of course, thank Wolfgang and Simon, who've been kind of working with me on this project, and you for your attention.
Great. Thank you, Constantin. We have lots of questions coming into the Q&A. Thank you. Again, a reminder for folks, if you do have some, can you make sure you put them in the Q&A? And we will turn it over to Davide at this point.
All right, thank you. And thanks, Constantin, for the very nice talk and overview. And if we can get this out of my way. Okay. All right, so yeah, so typically when I see, at least in statistics, these people asking or being tasked with being a discussant of the paper, I guess the first few minutes are always kind of like a recap or sort of like a a lesson learned from the paper, right? And given the topic of this paper, I thought it would be good to ask AI to do it for me. And actually, I just put the paper on Notebook LM, and it gave me a pretty good slide that I think, the AI itself titled Lessons for AI, which I think it's a pretty good summary of Constantin and Wolfgang and Simon's paper. So first of all, highlighting the power of good benchmarking, right? Testing against simple baselines is really essential to make sure that these more complex models are actually what is their added value. Simple models can also reveal the true nature and difficulty of the problem. And then, of course, better data and bigger models, it's not always the best. So I think, this whole stupid thing is just to say that, I think we can all agree that AI and large language models and foundation models in natural language tasks have been, showing very, very good results and impressive um, uh, impressive results. And this is not only true for large language models and for natural language tasks, but it's true in biology as well, right? I mean, I think we all have seen and been amazed by the results of the different version of AlphaFold for protein structure prediction. And even for sort of like medical imaging, I think there are very good successes of foundation models. And even though I would actually very liked, I would be very interested in seeing something similar to what they have done with benchmarking these large language models for sort of these sort of imaging tasks as well. It could be interesting to see if simpler models are able to achieve similar results in imaging as well. But I think there are some very good successes in biology for AI as well. So sort of the question of what is the performance of AI in single cell transcriptomics is a natural one, and I think it's a very timely and interesting question. And of course, as Constantin also mentioned at the end of his talk, After their paper came out, there are a few other papers essentially confirming the results that very simple baseline models can achieve similar or even better prediction performance than these sort of foundation models. And so what I've been sort of trying to ask myself is a little bit going into the why of this is happening. And I think another thing that I sort of stumbled upon on bioRxiv was, there's obviously not just this type of sort of prediction task in single cell omics. There's a whole different set of things that these sort of foundation model papers claim that their models are good at. Basically all the typical tasks that we do in single cell data analysis, kind of like batch effect correction or cell type annotation, gene network analysis, imputation, all of these different tasks. And there was this nice paper on bioRxiv that sort of gave a summary account of how well they perform in these different tasks. And you can sort of like, I know this is a dense slide, but you can kind of look at how in many of these tasks, overall, these sort of foundation models did not outperform more specific and very often simpler tools that are sort of specifically designed to tackle that problem. So I was, as I said, a little bit interested in trying to understand exactly what is going on and why these problems seem to be a little harder for these foundation models than other tasks that are very good at. And one thing that I thought about is what is actually the pre-training of these models, right? How are these models trained and on what type of data? And if you go through the publications of four of these foundation models, you see that they actually have a very sort of different set of data sets that they use for training. Starting from kind of like smaller training data sets, but very sort of well curated, to essentially all publicly available single cell resources existing out there. And you can already see that there are some non-trivial choices that these people had to make in sort of training these models, whether you only want to use normal known disease conditions or whether you want to include tumors and other type of diseases in your training. But the one thing that I sort of asked myself is, like, even for the biggest model, which was trained on 50 million human single cells, is this actually a lot of data? Is it actually a large data set, right? And what they say in that paper is that actually with about 20,000 genes and about 50 million cells, indeed you reach a magnitude of 1 trillion tokens, right? Which is comparable to the volume of the data that was used to train LLMs. But one question that I have is, like, is this actually true? What is the effective sample size when you keep in mind that many of these cells come from the same biological samples from the same individuals, and of course genes are correlated within cells, cells are correlated within samples, and so it is actually not clear, at least to me, whether these trillion tokens is the right unit to be counting for training these large models. And I also wanted to highlight one specific sentence by the paper we're discussing, where the authors say that the results that they show suggest that pre-training on the single cell Atlas data provided only a small benefit over random embeddings, but pre-training on perturbation data increased predictive performance. And so, in addition of how many cells, maybe the right question is what is the right data set to pre-train these models on? And another interesting paper that just came out on bioRxiv a few months ago is this Tahoe 100 million, which is a single cell, very large single cell perturbation atlas that may be actually a very good resources to train these models on. That said, there's also a few papers that suggest that bigger is not always better in terms of especially data quality, right? There's a couple of papers that show that increasing data set size and even data set diversity does not necessarily improve the performance of these models. There's this sort of idea of learning saturation. But I think a lot of us that work on single cell genomics or single cell omics know that there's a lot of artifacts and potential biases in this data that may indeed compromise performance of this model. And this on the lower side of this slide is an example of that. This is some sort of data in which there is clearly like a question of data quality. Some of these cells have a very low intron fraction, and in fact, people that reanalyze these data believe these are actually not single cell, but sort of like background RNA from cell debris. And in the original annotated data set that is used in some of these models as a pre-training, they are actually labeled as different cell states, right? So how does this matter, or does it matter for this large language model training? So overall, I guess a high quality, non-redundant data set for pre-training may be a very important milestone to achieve. And then I guess this is getting to the end of my slides. There's this idea that has been shown that this alignment between the training and loss functions sort of like specific for biologically motivated questions improve performance, which goes a little bit against this idea of these large models that can sort of like learn everything from the data. And then, this idea that simple embeddings are actually doing a lot already to represent the information that is available in single cell data. There's this paper by Quake that thinks about a cell like a bag of RNA, and they basically say already this could be thought about a 20,000 dimensional embedding. So do we need an additional embedding on top of that? I don't know. And of course, there's a question about complexity of the computational cost, not just for pre-training, but also for fine-tuning and inference. So just to conclude and maybe to spark a little bit of discussion, I thought about three sort of discussion points that I have for the speakers. First of all, I really enjoy their effort of, let's say, opening up the black box, right? This idea of the linear decoder of an LLM embedding really could be a way to understand what these embeddings are and what they are representing. And then, this idea of pre-training a linear model, I really liked it because it shows what is the actual added value of the pre-training compared to the model architecture step. So I don't know the thoughts about this. Then a more general question is it the right paradigm, this pre-training and fine-tuning for single cell omics, right? And if it is, what is, since we're in a bioconductor seminar series, what does that imply for repositories like Bioconductor? Should we move from a code repository to a kind of like model weights repository a la Hugging Face? And third, what is our role in this sort of AI world? I think benchmarking is a very good example of why we're still needed and we're not sort of like just surpassed by AI. And I thought another one would be uncertainty estimation. How do we know that the answer from the AI is actually any good? So with that, I'd love to hear the author's thoughts on this and uh, other things that I touched upon.
Yeah, thank you so much. I'm not sure. Robert, do you want to go?
No, I just was going to say thank you, Davide, and turn it over to you and Wolfgang and Simon to
Is it
rebut.
any useful to keep this up, or should we look at our faces?
I think it's useful to keep them. I think it's helpful to if you want to structure the thoughts about this. Well, thank you so much, first of all, for the kind of very kind summary and kind of extension building on our presentation and work. I think you're raising many interesting points there. I had to kind of smile kind of when you were mentioning the last point with kind of the role of the bioinformatician in the AI world. Like to what extent kind of do we kind of go for this kind of becoming like model developers ourselves in the AI world versus kind of trying to come along and kind of give critical feedback. But I do feel that by kind of benchmarking things, we're also learning quite a bit about how these models work, and maybe we can also provide ideas about kind of simpler things, and then kind of using ideas from AI, how to make them scale them up, make them more powerful, kind of integrate more data or have more parameters to learn. I think this would be something. So I feel like that I would try to avoid this division that we kind of only kind of critically evaluate what other people, machine learning people would develop.
I would, however, say that benchmarking is a really hard problem. I guess we have others may have already known this before, but in this project, I mean, we can realize how difficult it is to even decide what you benchmark for, like what your metrics are. And this is really a conceptually hard task. So I would like to sort of raise the visibility or the glory in this task a little bit. At some level, the benchmarking is probably harder than finding new architecture and coding and much of the other work, which is probably more mechanic, maybe easier to actually outsource to a robot.
Yeah, I think that's an interesting point with value of benchmarking, which I tended to undervalue in the past. In our work, I have even the feeling that we haven't really benchmarked much because we set up this simple baseline of just a linear model and showed that the models, that these all these advanced AI models don't even reach that. So essentially, if I'm slightly mean, I would say we have tried to let the hot air out of a hype. But as always, when there's a hype at the beginning, there is some substance below, which one then has to foster and it grows further. And as Wolfgang said at the beginning, we need to figure out what we really want to do with this model, what we want to use it for, and then we might get better at understanding how we benchmark it. Because in the end, the result, the work in practice is what counts. But I've recently read, I forgot unfortunately the reference, a nice AI opinion piece where somebody argued that the reason why other areas of AI made such great progress is precisely that they had clearly defined a reference data set on which everybody benchmarked their model so that you could quickly check whether some specific idea was actually progress or not. And in that sense, benchmarks really are also setting the test bed where people can check whether they are actually making progress. And hence, we can easily recognize that a small idea improves things a little bit. And here in this specific case, my feeling is, as David has said at the end, maybe it's not so much about the problem is, do we use the right kind of data, observable expression data or perturbation data? And now people make big perturbation data sets that might be useful. The other question is, how do we structure the training data? At the moment, we feed simply one cell after the other to this model, without even telling the model when one data set has ended and the next experiment starts. So of course it mixes all this together. So setting this test bed in which to test our models is not only coming up with benchmarks and metrics, but also coming up with good example data sets, with good ways to feed the data. And all these are obviously enough of work for us bioinformaticians to do. So for me, it made me happy that I won't get unemployed that quickly.
Okay, I will take the chair's prerogative and make a couple of comments, and then Wolfgang and Simon and Constantine, if you look through the 10 open questions, if there's any there that you want to address in words now, I would encourage, if you guys can, to go through them after the seminar so that we can get answers to them and post them so that there's sort of a record of how this went. And so I'll take a couple of tacks very quickly, I hope. So one, right, most of the studies that we look at are done in cell lines, and they're nice because cell lines are reasonably easy to manipulate, but they don't look like humans. And so part of the problem is we wouldn't expect them to look particularly like other systems that we might want to care about. So to Davide's point of, how much data do we actually have? Do we even have data on the system that we care about would be a question, right? And most of these cell lines, just for folks that don't work with them, are chosen simply because they are very permissive to transfection, and they are very resistant to, bad treatment and handling. And so they're not even representative of cell lines, let alone of humans. And then there's no genetic diversity here, right? So we have a field, bioinformatics. There's a fairly large cottage industry of trying to convince people that, EQTLs are a thing. And so if you don't have diversity of that nature and you're talking about predicting transcription, how does that come in? The one thing I was a bit surprised about in this and in others is we do have some genes that we know ought to affect transcription, right? Like transcription factors. And so I could think of a different baseline, which is if you can't see evidence of changes in transcription from things that we know ought to affect transcription, that should be a sort of a worse kind of a outcome. And it doesn't have to just be that, but phosphatases and tyrosine kinases and things of that nature that we know have fairly big impacts would be sets of genes that you would like to at least think of as true positives, and then there ought to be others that are true negatives. So I think there might be some room in there to think of benchmarks where we focus on specific genes where we might expect to see something as a possible way forward. And then the last one is just this sort of general idea that's not always clear. We sort of get stuck in thinking of large language models as big black boxes, but really all they're doing is estimating conditional probabilities, right? And the remarkable thing is that for things like ChatGPT, the conditional probability estimation of the next word in the sentence turns out to be remarkably useful. And we capture most of human language, no matter what language it's written in, with what I would call a relatively small models. And I don't know what the conditional expectation I'm trying to estimate here is, but there should be some kind of conditional expectation that we're trying to estimate. And maybe trying to be more explicit about what that is would help us understand how the models might work better. And in particular, what other features we might want to bring in to that. We might also be worried about the fact that ChatGPT is great because it's stochastic in nature. But not all large language models need to be stochastic, right? We're not trying to write an entertaining story. We're trying to do science. And how much stochasticity do we actually want in that particular process here? We may want a lot less than we're getting in some of these models. And so I will turn it back over to you guys, Constantin, Wolfgang, Simon, if you have more comments or if you want to pick some of the open questions.
So I found a question which I would like to comment on from Pantelis Vlachas, which is basically asking, could we use linear additive models to learn the residuals and then kind of build something on top of those residuals? And I think it's a kind of really good idea. And I think, yes, I want to just point out that there is some kind of work which basically is really taking this idea that if we kind of know such a good baseline, can we just learn the difference from that baseline? It's called salt and pepper. So if you look for a paper from Thomas Godelet, there is this idea, and I can also post a link, I think, later in the answer.
I can also maybe I mean, obviously the aim of the work was not to show that linear models are in some sense state of the art, or what you really sort of their baseline. And I guess I think we all hope that there's something more out there and that the foundation models can go beyond that. Yeah. I also sometimes think we have all of this tradition, or so at some level I think the Go categories and gene signature databases, or even string, those gene network databases, they try to do something that we now try to use foundation models for. So maybe some of those use cases that we're now trying to do, like for instance unseen gene function prediction or phenotype prediction, is also already contained as a seed, at least in these prior gene network approaches. So one of the tasks I see, one of the opportunities I see with these foundation models is basically something that learns all the biology and sort of contains it in some sort of elegant, hopefully, and comprehensive way that would imply, for instance, knowing what is a transcription factor, what is a phosphatase, and what each gene is doing.
Yeah, that sounds like an important point, because the success of large language models is they don't start from scratch. They build on all our work that we humans assembled over many centuries, because they've read it all. And here with the single cell things, we basically ask these models to start from scratch. We give them a bunch of observational or maybe perturbative data. We don't even give them the opportunities to do experiments. We don't even give the models the metadata about what perturbations have happened in the things. It's basically expression values without any context, and we're throwing away many decades of work of biology. If this would work, that actually would be quite depressing, because then all our previous work was useless. So I'm a bit happy that it seems that we have to actually, if we want to use AI, we have to teach AI about all the stuff we already have figured out about biology.
Yeah, so maybe we're just going to continue to go through the questions. So the most upvoted question is by Thomas Sandmann, which basically asks us to speculate a bit what would be the ways that we could kind of use to improve the predictions. Is it better data? Is it less noisy data, different data types, better algorithms? It's probably going to be a mix of all of it. I think we have already kind of touched on some of them, but just so I personally think I see some evidence in our paper as well that training on perturbation data is really helpful instead of just training on just observational data. So having something more pertinent, some more pertinent data. I would also kind of coming back to Davide's point about the kind of cells as sentences and kind of genes as words, which is the typical architecture that currently the transformer-based foundation models are using. It feels a bit odd personally to me. I think there's some justification. So they say that, well, they're looking at the expressed genes, which roughly corresponds to these 3,000, and they use a ranking and how much information do you really lose by ranking things. But it doesn't feel like a particularly elegant architecture. It feels like something that was basically transported straight from kind of natural language processing to single cell processing. So I could imagine that maybe more bespoke architectures for single cell processing also would really make a big difference. However, this is of course much more difficult to benchmark. And I guess the only real proof that this is true is basically somebody building it and then showing that it really does make a big difference in an ablation, for example, type of benchmark. Yeah, I guess less noisy data always good. I'm not sure. I find it difficult, to be honest, to say, because we are here looking always at the mean. Like we have like hundreds of cells which actually have a perturbation, and then we just take the mean of that to then calculate any type of metric on that. And that already gets rid of quite a bit of noise. Of course, if you have too few cells with a particular perturbation, then noise really does become a big problem. And if you look at very lowly expressed genes, also noise becomes another problem. I think I don't have a particularly strong opinion on the existing data that this might be the main.
So, can I make one small comment here, Constantin? So what's interesting is some of the people that talk about these foundation models call them virtual cells.
Yes.
And this thing has actually has a long history. There were already virtual cell projects 20 years ago, but at those times, it wasn't AI, it was basically big systems of ordinary differential equations that people wanted to solve and model a cell. Uh, and and you can then think maybe a big system of partial differential equations also have space and so. So, and of course now the pendulum has swung and we have sort of given up that idea, and now it's a big transformer, deep neural network. And maybe the way forward is somewhere in between, or even something else that contains some physics-based ideas plus this more information theoretical approach.
I think there's one also interesting question from Paul Harrison asking if we could design now a better machine learning model now that we have established these metrics and these tasks. It does say, like, can you design better machine learning model? I don't know if I personally can, or if it's actually that's what the question the person asking them means. I would be curious to give it a try. So of course I have some ideas for myself, like what would be maybe interesting avenues to explore. But I think what kind of we have seen is that in particular the double perturbation prediction task seems to be really hard. Like even if you look at some of the models that came out more recently, even those models seem to really struggle making meaningful progress on this double perturbation prediction task. Maybe because also it is just so much more difficult to find a good representation how these different things interact. My feeling is that there might be still some low-hanging fruits, but in particular, kind of for predicting, for example, buffering effects versus, yeah, non-additive ones. So identifying the buffering non-additive interactions. I think in particular synergetic interactions, so where kind of the gene expression for a downstream gene becomes even more with the double perturbation. That seems to be really hard, and I don't know of an obvious way to kind of make good progress on that. And if just throwing it in a kind of gradient descent type of algorithm will really get us much forward there.
All right, I'm going to call time. I want to really thank our European colleagues for staying up so late at night and giving this talk. We tried to find a time that would include as many people as possible, and the attendance was quite high, so I really appreciate it. So thank you, Simon, Wolfgang, Davide, and Constantine. I also would like people to, thank Erica, who did all of the heavy lifting for this seminar and got folks organized, me in particular. And she will be sending out a post-seminar survey. Please do respond to that. We don't know what format we're going to end up with, but if all of them get good questions and good audiences like this, then I think we do have something that looks to me like a fairly nice formula for trying to, have a good interactive bioconductor seminar. So thank you all for coming. And I guess that's everything. Anybody else have a closing comment on our panel?
Maybe I just briefly mentioned that I have a few links to papers in my slides, so I'll post them on Zulip.
Thank you. All right.